Hello, I am Ambarish and I will be discussing about CYP450 Amplichip test today. Successful pharmacological treatment implies effective therapy without the development of adverse drug reactions. Effective therapy entails a beneficial response to treatment despite the inherited and acquired variations in drug bioavailability and targeting that often lead to treatment failures, even for compliant patients. ADRs and TFs represent a significant burden to overall health costs. Indeed, more than 4.7 billion serious ADRs occur annually in the world, resulting in more than 3.2 million deaths and costing the economy an estimated of $750 billion. Moreover, many patients receiving prescription drugs fail to fully respond to first line treatment and require additional pharmacological interventions, further escalating these costs. Factors contributing to ADRs and TFs include drug-drug interactions, physician-patient miscommunication regarding proper drug administration, physician prescription error, and significant differences in how individuals metabolize drugs. Genetically encoded variations in the activity of drug metabolizing enzymes has emerged as a potential tool for predicting drug response and ADRs in populations over the year. Understanding the links between genetic polymorphism of drug metabolizing enzymes and responses to therapy may lead to individualized medicine. Among the drug metabolizing pathways, the CYP450 system represents the main component of phase 1 metabolism. Genes encoding drug metabolizing CYP450 enzymes such as CYP2C19 and CYP2D6 are highly polymorphic. Predictions of an individual's capacity to metabolize pharmaceuticals based on the genotype of an polymorphic CYP450 enzyme is now possible. This has the potential to significantly decrease ADRs and TFs in the future. However, introduction of this approach into clinical practice requires robust, affordable, and accessible genotyping technologies that can be easily performed by most clinical laboratories and can be applied to large populations undergoing therapy. The Amplichip CYP450 test is the first microarray based test to be approved for diagnostic use in the clinical setting. Based on the Affymetrix gene chip platform, the Amplichip CYP450 test assays phenotype of drug metabolism capacity for the CYP450 genes CYP2D6 and CYP2C19, which are involved in the metabolism of several commonly prescribed drugs. Both the Amplichip test and the Affymetrix platform received US FDA approval. The Amplichip CYP450 test may allow pre-prescription decision making based on a patient's capacity to metabolize certain drugs and may help prevent ADRs and TFs by providing a basis for adjusting dosages or prescribing alternate therapies when available. However, the Amplichip CYP450 test is applicable to only a select list of drugs, representing almost 37.5% of currently prescribed medications. Perhaps the most exciting and potentially important aspects of this technology is the FDA's approval of the Affymetrix platform on which the Amplichip test is run. The Affymetrix system is capable of running any number of specific chip-based assays and is highly scalable, providing the means for future versions of the Amplichip to assess variations in a greater number of drug metabolizing genes. The tests are simplified, semi-automated, and applicable in small laboratories, obviating the need for specialized molecular genetic laboratories. The test analyzes the DNA of a patient to determine the genotype, and predictions of the phenotype can then be made. The DNA sample comes from blood or alternatively comes from buccal swab, and the analysis has mainly 5 steps after DNA is extracted from patient sample. First, PCR amplification of the gene, fragmentation and leveling of the PCR product, hybridization and staining on the Amplichip DNA microarray, scanning the chip and analyzing the data. Specifically, the Amplichip CYP450 test is built around the well-proven Affymetrix gene chip platform. Briefly, highly dense fields of oligonucleotides are chemically synthesized on glass chips using a series of photolithographic activation steps, a technology adapted from the semiconductor industry. The sample is hybridized to these fields of oligonucleotides and results are obtained using an automated scanner and displayed on a computer screen. The first application of the Affymetrix technology was in the quantification, of large numbers of expressed RNA transcripts using in vitro transcription to produce suitably level targets, that is, gene expressing profiling. But the Amplichip CYP450 test uses the much more stable nucleic acid DNA as a template for labeling and involves a polymerase chain reaction based on Roche's well-proven platform. The Amplichip CYP450 test provides predictive phenotypes of the drug metabolism capacity of the CYP450 genes CYP2D6 and CYP2C19. To do this, it analyzes 29 polymorphisms and variations in the highly polymorphic CYP2D6 gene and 2 polymorphisms in CYP2C19 gene. Numerous genetic and non-genetic factors can affect drug response in an individual. Indeed, inherited differences in the way a person metabolizes drugs may afford a basis on which to predict treatment responses. For example, ADRs and TFs often occur when the therapeutic window is narrow and thus only a small margin exists between effective and toxic doses. Knowledge of an individual's capacity to metabolize such drugs before initiations of therapy could provide a way to tailor medication doses to achieve the optimal effect while avoiding toxicity. Alternatively, this knowledge may suggest the use of alternate therapies if applicable. Clinical relevance transpires when variation in human CYP450 metabolism is relatively abundant and individuals are treated with commonly used medications. In such cases, the potential for notable clinical consequences due to wide variation in metabolic capacity exists. In addition to the clinical consequences of varying CYP450 metabolic capacity in the population, the resulting ADRs and TFs can have a significant impact on treatment costs. Most studies of the effect of CYP2C19 polymorphism on drug metabolism have focused on the PPIs because these are among the most widely used medications metabolized by this enzyme. Traditional triple therapy for Helicobacter pylori eradication involves administration of PPI such as omeprazole or lansoprazole in combination with two antibiotics, generally amoxicillin and clarithromycin. This treatment is widely successful with complete eradication achieved in greater than 80% of the individuals. Interestingly, studies have shown near complete Helicobacter pylori eradication in patients with CYP2C19PM phenotypes using omeprazole in combination with dual and triple therapy schemes, suggesting that clarithromycin may not be needed as a first-line treatment in these individuals. Individuals who are homozygous for the wild CYP2C19-1 allele are most likely to fail this first-line treatment effort using standard approaches because the PPI is quickly metabolized and sufficient acid inhibition is not maintained. Recent studies have demonstrated improved treatment efficacy in this EM group from various alternative therapies, including the concomitant dosing of 2 PPIs and administration of high dose of 40 mg approx esomeprazole, both in combination with standard antibiotic regimens. Avoidance of TMs could have a significant clinical impact by preventing complications of prolonged H. pylori infection and minimizing the development of drug-resistant strains from excessive use of broad-spectrum antibiotics. Apart from that, since a lot of the CYP2D6 substrates are psychiatric drugs like antidepressants, antipsychotic drugs, the Amplichip CYP450 has been extensively used in psychiatry over the last few years. In spite of different critical conditions where it has been proven that this particular test can be very applicable, the main criticisms of this test is that the test finds out the genotype of the patient, which does not necessarily cover all the phenotypes. For example, some argue that the so-called ultra-rapid metabolizer who has extra copies of the 2D6 gene expressed cannot be reliably tested. Also, the test does not cover some rarer genotypes or genotypes that have not been discovered yet.